Hey team. Well, guess what? The last lecture. That's right. And it's been an interesting semester. I've never taught a botany class quite like this before, obviously. I'm glad that you made it through. I hope you've, learned and that it's been meaningful. I realize that it's always going to be better when we can be together and, actually be a little more involved with each other's learning, but we did the best we could. I hope you've enjoyed the class. And so today we will finish up and thanks for attending. Thanks for watching all these videos. I know it's not the easiest way in the world to learn, so I do appreciate your patience. Thank you. So what we have left, just to be clear, is to study the angiosperms, the flowering plant life cycle. And think about it, we've come full circle. We started out talking about plants at the beginning of the semester. We talked about plant morphology, tissues and plant organs and all that. Well, now we've gone through all those other kingdoms of living things, the fungi and the protista and the bacteria and all the others. And now we've come back to plants in order to understand a little bit more about their life cycle, primarily. So we're gonna, spend most of this lecture will be on plant life cycle. It's a fascinating life cycle due to a phenomenon called double fertilization. And you're gonna see this, it's very strange and interesting. Okay, so just before we do that, let's take a second to look at a little paleobotany here. Paleobotany is, plants of the past, you might think of it as, and kind of when they lived on the earth is an interesting thing to think about. So here you can see, here are the different epochs or eras of periods, if you will, of geological time. Sorry, it's having a hard time saying that. So, you can go all the way back to the Silurian, 444 million years ago. We'll say 450 MYA, million years ago. And then, when we're going in this direction, here's the present, okay? And so it's nice to have a sense of what grew when on the earth in terms of plants. And you can see the algae have been around a really long time. They've been around since the Silurian. We believe that somewhere between 300 and 250, somewhere in the neighborhood of 400,000 years ago, the first land plants evolved somewhere right around in here. And those would have been the mosses, right? And so, the mosses really did well and continue to do well on Earth. Let's look at the early vascular plants, primarily ferns and their allies. And so what you want to realize is that during this period, the Carboniferous, this is really a moist and wet time, warm and wet, I should say, time on the earth. And so this is when the ferns and their allies had their day, okay? By allies, remember those other groups, the Lycophyta, the Splenophyta, the horsetails. And so when we burn coal, we're actually burning horsetails and lycopodiums and ferns from this Carboniferous period, which was, between 350,000 and 350 million and 300 million years ago. Now look at ginkgo. The ginkgos get their own line here. And you can see a bit after the ferns, right around the Triassic, the ginkgos began to dominate the flora or become part of the flora. And like I said, there are thousands of species of fossil ginkgo. And so, ginkgo biloba is the only species left, but there are lots and lots of fossil members of the ginkgo clan, okay? You can see conifers right here. As we get up into this period, into the Cretaceous and even the Upper Jurassic, what we see happening on Earth is a cooling and drying. And this is really when the conifers dominated the Earth. And then we have the first seed plants, the cycads rather, coming in after that. And then notice when the flowering plants come in, they don't come in until very late in the game. And honestly, this says 130 million years ago. Really, it was about 80 million years ago that they became widespread and diverse and dominant on the Earth. Now, there's this big hole in the fossil record here. We don't have any good transition species, really, between the gymnosperms and the angiosperms. And this is very frustrating to paleobotanists, because you would expect there to be some transitional species, but there seems to be a big hole in the fossil record between these two groups. It's as if the angiosperms simply appear in great numbers and great diversity right around 80 million years ago. So one of the theories that scientists have come up with is called the upland theory, to try to explain, why in the world don't we have intermediary species in the fossil record. The upland theory says that flowering plants evolved on upland slopes in mountainous regions. And this sort of habitat isn't really conducive to fossilization, because, in order to be fossilized, you got to be buried. And so it wasn't until the plants started to colonize lower down, and then there could be a landslide and you would have fossilization occurring. To me, this is a little bit of a weak theory, but it is the theory, the current working theory about, why we don't have this transition. Okay. So that just gives you a little bit of an idea of what grew when on the Earth. And of course, what dominates the Earth now? Well, angiosperms, flowering plants, are the most dominant species on the Earth. And so we might take a second to think about, why that is. Well, why that is that they have evolved so quickly to fill pretty much every niche or habitat on Earth. Now, granted, there are places where conifers dominate, but for the most part, there's an angiosperm for every habitat on Earth. How do you go from a cone to a flower? That's the next thing that we want to think about. How do you go from a gymnosperm to an angiosperm? Angiosperm, angion, this word here, angion means vessel. So these are the vessel seed plants, right? What's the vessel? What surrounds the seeds in an angiosperm, in a flowering plant? The vessel is the fruit, right? So how do you go from a cone to a fruit? Well, if you think about those bracts on a cone, and let's say my hands represent one of those bracts of a cone with their seed here and a seed here, really all that needs to happen is an enrolling of that little bract around those seeds. And then all of a sudden, now we can call that a fruit. It's surrounding the seeds. And so we believe over evolutionary time, this occurred. One of the other really dramatic and beneficial things that occurred with the flower was the introduction of vector pollination, right? All conifers are wind pollinated. And with the advent of the flower, now you have this fancy showy thing to attract insects to move that pollen around. And this would be considered an evolutionary advancement, obviously. Now you don't have to make copious amounts of pollen. You can just make a little bit and depend on the vector to move it effectively, rather than having to make huge amounts and using the wind. Okay. So that's all good stuff to think about as we enter into the next unit. Okay, so this gives you a little bit of information. Now here's the good news for taxonomy. All flowering plants are put in one division. So although the spore plants and the gymnosperms had lots of divisions, we put all flowering plants in the Magnoliophyta. If it makes a flower, it's in the Magnoliophyta. Most abundant and ecologically successful plants on Earth. We don't know how many species there are, probably upwards of 300,000 species. And, you know, the flower we would consider an evolutionary advancement over the cone, and vector pollination an evolutionary advancement over wind pollination. Now, here's where we have to be careful using that word advancement. When you see that word, it has this connotation of being beneficial, right? Or you think it, more primitive versus advanced, you kind of think in your mind, well, advanced means, you know, better able to survive, but that's not really what it means. And we're going to see the most highly evolved, or you might say advanced flowering plants are the orchids. And we're going to learn that that's really not, ends up not being much of an advantage at all. So be careful getting this idea of evolutionary advancement mixed up with survival rate or ability. Okay. This gives you an idea of, you know, some of the terrestrial orchids, and we consider the orchid to be, you know, the sort of the pinnacle of evolution of a flowering plant. We'll talk about why that is in just a bit, and we'll talk about primitive versus advanced traits. But before we do that, let's do the life cycle. And so again, you know, we kind of started the life cycle before, remember, because we talked about the flower and the flower parts, and we said, you know, inside the ovary there are ovules, and the ovules have eggs, but that was as far as we went. And so now we're going to do this in much greater detail, and we're going to see actually how this works. So this image is really nice. It shows the male, right, formation of the microgametophyte, which ends up being the pollen grain here. There's mitosis, and there's a pollen grain. And then it shows the female, the megagametophyte, okay, megaspore, etc. But let's go to our fancy whiteboard, Look how fancy this is. And while it's up here, you know, I'll just go ahead and tell you, this is going to be the same. All of this is going to be the same for the male, except where it's taking place is in an anther instead of a strobilus. Okay, it's also going to be very similar for the female, so that's all good. So let's think about a flower. Let's remember the flower parts. Okay, sepals, petals, stamens, and then a pistil in the middle. Okay, and remember, inside a pistil is one or more ovules. And you know, that's, I hate to have to do this, but I'm going to have to draw this again because I didn't really draw it quite big enough. Let me make it a little bigger. It'll be easier to see what's going on if I make it bigger. My fabulous artistic ability. There's the petals. What I really need to draw in here is the pistil because there's a lot going on here. Normally I would have a big whiteboard to do this. So there's the ovary wall. I'm going to draw the stamens in, don't worry. And then protruding into the ovary wall is one of these ovules, and inside the ovule is a cell, okay? And that cell, students always want to call it the egg, and I can see why. But keep in mind, this is of the sporophyte, right? The flower is part of the sporophyte. The flowering plant is a sporophyte. So we have sepals, petals, and then this becomes important, the anther, because that's where we're going to make the male part. And then the pistil with its stigma, style, and there's the ovary, okay? And then what is inside the ovary, this thing in here, is an ovule with, don't call it an egg yet, it is a megaspore mother cell. And it's got to be 2N because we haven't done meiosis yet. So remember, it was basically the same thing that we saw in the cone, except it was happening in a cone instead of an ovary. Okay, so let's remember what these parts do. The sepals protect the bud, the petals attract pollinators, the anther becomes important now. Remember, this whole thing is called the stamen. Sorry. I know it's hard to see all that. And then that consists of the anther and the filament, right? Okay, so let's look what happens in the anther. If you look at the anther and you cut it in a cross section like this, it has these 4 chambers, kind of like this. And inside those chambers, I'm going to do them in yellow because yellow is the color of pollen. Inside those chambers, there are cells. And guess what we're going to call those cells? Microspore mother cells. And what's their ploidy? They got to be 2N because we haven't done meiosis yet. Okay. Well, that's what they're going to do, right? They're going to do meiosis. I'm going to put MMCs for microspore mother cells. And then what you're going to get, I'll just draw one side of it, are tetrads, right, of microspores inside of this chamber of the anther. I'll draw a few of those in and label them. Okay, and now we're haploid. The microspores are 1N. They're the products of this meiosis that occurred. Okay, so now what happens around this stage is those microspores come apart and become individual cells, single cells, and the anther actually kind of splits open and releases them usually around this stage or a little further. So let's now draw one microspore. It's just one cell, one microspore. It's haploid. It does the same thing that it did in the conifer. Mitosis. And we're going to get 2 cells. Remember, this is called a generative cell. And this is called a tube cell. And then one more picture. Hopefully y'all can see this. I don't know where my picture is, somewhere on there probably. We do another mitosis to make 3 cells. 2 of them are sperm. And the third is the tube cell. Okay, and just to label this, this is mitosis. Oops, come on now. What is happening? Okay, sorry about the color. Okay, so what is this thing? Well, you call it a pollen grain. I call it the microgametophyte. What's its ploidy? It's got to be haploid. It's the product of these microspores, which are the product of meiosis. Okay, so now we have a pollen grain with its 2 sperm. This is the mature microgametophyte. Again, the microgametophyte has been reduced down from this free-living thing like we saw back in Ulva a long time ago, all the way down to this 3-celled structure. Okay, so now let's look at the female. Okay, if we draw, let's just draw one of these. Let's just draw the ovule kind of big, because we need to label all this stuff. There's a bunch of terminology. There's the megaspore mother cell. These are called integuments. And the integuments are these little sort of bracts here. And you'll notice that there's an opening right here in between them. And that opening is called the micropyle. A lot of terminology here. So the integuments are these little bracts. The integuments actually end up becoming the seed coat or the endocarp of the fruit, depending on who you ask, okay? But you'll notice they don't quite touch on either side, these little bract-like structures. And there's an opening called the micropyle inside. And then what is this cell? It is the megaspore mother cell. We're still diploid, okay? Well, look what happens. Meiosis occurs, and we get a linear tetrad, just like we did before. 1, 2, 3, 4. 3 die again, like they did before. 1, 2, 3, and this one at the end survives. Now, here's the tricky thing. Let's make it different color. Let's make it a different color to remember the ploidy. Let's make this one blue now. And blue kind of represents haploid, because this cell is haploid. This is still not an egg. I know you want to call it an egg, but it's still not an egg. We go from megaspore mother cell to what? What are these 4 megaspores? Haploid. 3 die, and we're left with 1 megaspore. Okay. Now, here's a weird thing that happens. I know I keep saying stuff's weird, and this is weird. We're going to do mitosis times 3. Now, by mitosis times 3, I mean this one cell does mitosis once, and you get how many cells when you do mitosis? You get 2, right? So one cell makes 2, that's the first mitosis. 2 cells make 4, that's the second. And then 4 cells for the third mitosis make 8. And so we end up now with this 8-celled structure. I'm going to draw it nice and big so we can fit them all in. Eight cells in here. I'm going to do them all in blue because they're all haploid, right? Here's how they arrange themselves. There's one really big cell there, and 2 on either side. There's 2 in the middle. And then there's 3 up here. Okay, and of course, all these cells have names, and they arrange themselves in this order. So we're going to go ahead and label what these cells are. This big one, you could guess, what do we need? What's the female gamete? This big one is the egg, right? It's haploid. The ones on either side of the egg, these 2, are called synergids. Weird name, I know. There's 2 of those. Be careful, I don't want you to think those are diploid. Let's write the 2 there, and let's erase this. Darn it. Let me just have to redo it there. 2 synergids. Okay. They're haploid. All this stuff's the product of meiosis. Now, these 2 in the middle are called the polar nuclei. Makes no sense to me. Why does that not make any sense? Where are the poles at the ends? Well, the polar nuclei are in the middle. There's 2 of those. They're haploid. And then we have these 3 cells, which are called the antipodals. 3 of those, and they're haploid. What is this structure? It is the mature megagametophyte. So now what we see, like we've been saying, is a reduction in the size of the gametophyte generation in plants from the algae, through the mosses, through the ferns, through the gymnosperms. And now when we get to the angiosperms, we're dealing with a gametophyte generation. That is 3 cells in the microgametophyte, or the male, and 8 cells in the female. And so we see a dramatic reduction. The trend, it appears, would be going toward not having a gametophyte generation at all, which is what animals have, right? Do we have a gametophyte generation? No. We are sporophytes. We are diploid organisms that generate gametes. We don't go through that gametophyte generation to generate a gamete. Okay. So a lot of terminology here. I would make a vocabulary list and put these words on it: integuments, micropyle, synergids, polar nuclei, antipodals, generative cell, microgametophyte, megagametophyte, microspore mother cell, et cetera, et cetera, et cetera. You need to know this terminology. I'll tell you, this last test, y'all, there's not a lot of info on it, and so you really got to know it frontwards and backwards, because I'm struggling to get your 500 points out of the several lectures that we've done. Okay, so now look, we got sperm, we got an egg, and what can we do? We've done meiosis twice, once for the male and once for the female. What's the other hallmark event of sexual reproduction? We got to do fusion to get back to sporophyte. So I'm going to erase this, take one last look at it, and then I'm going to erase it. And we're going to draw and see, oh, what's about this double fertilization business. Okay. Bye-bye. Okay, so now check it out. I'm going to draw this giant ovary, really big. Okay, and I'm going to draw a giant ovule inside of it. Aren't y'all glad I took art? I didn't take art. I know I'm not the greatest artist in the world, but I'm trying my best. Okay, there we go. And let's remember what this stuff is: stigma style, and this is the ovary wall. Here's the ovule, and now look, I'm going to draw in. I'm going to draw in my cells. And, you know, there's the important one, the egg in the middle. But we're going to see these 2 polar nuclei end up being important as well. As far as what these other cells do, the antipodals, nobody really knows what they do, y'all. They're just there. The synergids, we guess, are some sort of, there's 2 theories. One is that the synergids act as sort of like a beacon to guide the sperm to the egg. So they live on either side of the egg, and they send out maybe some sort of chemical signal to attract the sperm. But we don't know for sure. We also have a theory that they may provide nutritional backup to the egg to keep the egg alive for a longer period of time. We just don't know for sure. Okay, so how does this thing start? Well, it starts with compatible pollen grain of a compatible species landing on the stigma. And remember, the stigma can be hairy or sticky to attract the pollen, or maybe a bug brought it, okay? Now, as long as it's compatible, it will actually germinate. If it's the wrong species, it will not germinate, it'll just sit there. But if it's the right species, it'll germinate. And by germinate, I mean this tube cell begins to produce these digestive enzymes, and it literally starts burning its way through the female tissue like this. And it goes through the ovary wall, but it stays in the wall of the ovary. It goes all the way around to the bottom, which is it's taking a circuitous route. Okay. This is a tube of cytoplasm. Try to wrap your brain around that. Okay. And it keeps going. It's attracted to the egg. It goes in between the integuments through the micropyle and makes a tube of cytoplasm all the way, continuous tube of cytoplasm all the way between the sperm and the egg. Now, what then happens is pretty crazy. Both of these sperm migrate they kind of race each other down the tube, believe it or not. The sperm are just nuclei at this point. They're moving through the cytoplasm toward the egg. And I like to think of it like the first sperm, the lucky sperm, fuses with the egg. And when that sperm and egg fuse, the first cell of the next generation is formed, and that is called the zygote. Now we're back to diploid. And you'd think it would end there, but it doesn't. What happens then is the other sperm, I guess maybe feeling left out, migrates past the zygote to this region of where the two polar nuclei are. And are you ready for this? All three of them fuse together. What the heck are we talking about? All three of these haploid cells fuse. And what they form is something unique to life cycles of living things. What they form, fusion of, let's be clear, let's just hop back down here real quick. This was the sperm and the egg, both one end, right? This was polar nuclei, two of those plus a sperm makes 3N tissue. This is naturally occurring triploid tissue that we're talking about here, okay? Think about the implications of that. It's really pretty wacky, okay? But this is what happens. And this cell is called the endosperm cell. And it's triploid. This one cell here is triploid cell. The antipodals just hang out. They don't really do anything. So how weird is that? This phenomenon where one of the sperm fuses with the egg and the other with the two polar nuclei is called double fertilization. Now, at this point, when this double fertilization event has occurred, lots of changes begin to happen, right? For example, what is the fate of all of this tissue and what is the ploidy of all of it? If you think about this one drawing, think about the ploidy of everything. For example, what's the ploidy of the ovary wall? It's of the sporophyte, it's diploid. What's the ploidy of the synergids and the tube cell nucleus and the antipodals? They're all haploid. They're products of meiosis, right? What about the zygote? Zygote's always diploid. And then we have this triploid endosperm cell. So in this one drawing, we have haploid cells. We have diploid cells from the sporophyte. We have a diploid zygote that will become the next generation. And we have haploid cells. Okay, pretty wacky. What's the fate of all this tissue? Upon fertilization, what happens to the ovary? The ovary becomes the fruit, right? The ovule becomes the seed. The zygote, through mitosis times many, becomes the embryo. In an embryo, just like in an animal, a plant embryo is a perfect little baby plant. Isn't that cool? The integuments become the seed coat. Dun, da, da, da. And the endosperm cell, which is triploid, becomes the endosperm. And what the endosperm is the nutritive tissue that keeps the embryo alive throughout its dormant period waiting to germinate and grow into a new plant. Okay, so think about a peanut. If you take a peanut, you know, it's a legume, right? So you know what the fruit is. It's the dry shell when you crack it off. The seed coat is that red skin. That's the endocarp of the fruit or the seed coat of the seed. But think about if you take a peanut and you crack it in half,